Hello everyone, welcome to the Zimbabwe lab at the Cambridge Stem Cell Institute. My name is Francesca and I'm one of the senior authors of the paper. I'm going to give you a little bit of background of our study. Inflammatory bowel disease refers to a group of disorders characterized by the chronic inflammations of the gastrointestinal tract. It includes Crohn's disease and ulcerative colitis. The epithelium of the inflammatory bowel diseases patients has been characterized by many alterations that are crucial for the pathogenesis of the diseases. In the IBD, the epithelial barrier becomes compromised, often resulting in increased permeability of the intestine, and these disruptions allow laminar contents, including bacteria and their products, to penetrate the submucosa, triggering inflammatory responses and immune responses. A novel in vitro model to study this disease is the human-derived intestinal epithelial organoids, which are three-dimensional structures that mimic the architecture and functionality of the intestinal original tissues. This model provides a reliable and controllable environment where it is possible to study the disease mechanisms and its progression. Epigenetic mechanisms, including DNAm methylations, have been proposed to play a key role in Crohn's disease pathogenesis. DNAm methylation is an hereditable and stable mark which underpins cellular identity and involved in many cellular functions. So its alterations lead to diseases. Since the inflammation tends to recur to the same anatomical site in Crohn's patients, even after treatment, we hypothesize that this recurrence might have an epigenomic origin. Therefore, we focus our attention on investigating disease-associated DNA methylation changes in intestinal epithelial cells to understand their contribution to Crohn's diseases. We generated a biobank of 312 human-derived organoids from the mucosa of 168 patients with Crohn's disease, ulcerative colitis, and also from healthy controls. We performed a genome-wide molecular profiling, including DNA methylation, bulk RNA-seq, and single-cell RNA sequencing. I'll hand over now to Rachel, who will explain us how these DNA methylation changes have been analyzed and our main findings.
I'm Rachel Edgar, one of the first authors of the paper, and I'll describe some of the computational aspects of our work. So in studying IBD, we had some expectation that DNA methylation, as it's a relatively stable mark, could be a good biomarker for Crohn's disease. So looking at genome-wide DNA methylation, we found that MHC-I genes, in particular the transcription factor NLRC5, were consistently less methylated in Crohn's patients. This lower methylation was retained in patients over time, and importantly retained within patient-derived organoids. So in these organoids, we also could see that MHC-I methylation was associated with Crohn's disease severity. And using a machine learning approach, we were able to stratify patients by disease severity based on a prognostic signature of only 28 CpGs. So having seen that the lower methylation in Crohn's was associated with higher expression, we wanted to explore if this expression differences were cell type specific. So we're able to use single-cell transcriptomics in both the organoids and patient biopsies, and we found that MHC-I higher expression in Crohn's was consistent across epithelial cell types and even seen within crypt epithelial stem cells. I'll now hand it over to Tom to explain some of the experiments to understand the functional mechanisms of this MHC-I dysregulation in Crohn's.
Thanks, Rachel. I'm Tom, and I work in the Milner Therapeutics Institute in close collaboration with the Zilbar Group. We were next able to validate our findings using in vitro and in vivo studies. Firstly, we used genetic engineering techniques in organoids to dissect the MHC class I pathway. These findings confirmed NLRC5 to be a key transcriptional activator in this context. We were also able to demonstrate active function of this pathway in this cell type using co-culture experiments between organoids and CD8 T cells. We were then able to confirm the wider clinical relevance of this gene by exposing NLRC5 deficient mice to the DSS model of colitis. These mice were found to be almost completely protected from colitic symptoms. Finally, we tested the stability and intrinsic nature of the disease-associated changes we had found by functionally testing organoids selected from our organoid biobank based on DNA methylation profile. Organoids hypomethylated in NLRC5 were found to have altered MHC class I response to interferon gamma. I'll now hand back to Francesca.
Thanks, Tom. In summary, our study has identified epigenetically regulated intestinal MHC-I as a novel mechanism in Crohn's disease pathogenesis, suggesting that epithelial cells play also a role as a non-classical antigen-presenting cell type. In addition, organoid-specific MHC-I methylation changes correlates to the clinical outcomes, highlighting the potential for the development of clinical biomarkers. Thank you for listening, and we hope you enjoyed the paper.